Hello, my name is Joshua Mugavi. I welcome you to Membrane Transport. Lesson objectives. Today we shall look at membrane transport, diffusion, facilitated diffusion, osmosis, osmolality, active transport, vesicular transport, and bulk transport. You are welcome. We shall start with the types of membrane transport. Membrane transport occurs in one of two processes. It occurs in one of two processes. The first process is the passive process. Passive process. The passive process, this one involves the movement along the concentration gradient. Movement along the concentration gradient. Now that one means that substances are moving from a region of high concentration and they go to the region of low concentration. When substances are moving along the concentration gradient, what we mean is that they are moving from a region of high concentration going down to the region of low concentration. That one is what we call passive transport. Does not involve expenditure of energy. In the passive transport, there is no involvement of expenditure of energy. We don't use ATP when we are using passive transport. Another one is active transport. Now, active transport, this one involves the movement against the concentration gradient. Substances are moving against the concentration gradient. Now, this one, because they are moving against the concentration gradient, it means that now the molecules or the substances are moving from a low region of low concentration to a region of high concentration. And that one is what we say as active transport, or moving against the concentration gradient. What do we mean by active? Active means that there is expenditure of energy. We have to expend or we have to use energy. So if we are moving against the concentration gradient, it means that we have to spend some energy, and that spending of energy is the one that is going to help us to move against the concentration gradient, moving from a region of low concentration to a region of high concentration. We are moving against the concentration gradient, and that one is referred to as active transport. Now, we have already talked about that this one involves the expenditure. There is the expenditure of energy. So we end up using ATP in active transport. Particles move across the membrane by simple diffusion. They can move by facilitated diffusion. They can move by osmosis, and they can also move by active transport. That's how materials or particles move across the membranes. Simple diffusion, facilitated diffusion, osmosis, and active transport. Types of passive transport. We want to look at the types of passive transport. There are three main types of passive transport mechanism. We have three main types of passive transport mechanism. Those are the ones we want to look at right now. Now, we shall start with the simple diffusion. In simple diffusion, small, or sometimes what we call lipophilic molecules, these ones are going to move freely across the plasma membrane. Those small molecules can move freely across a plasma membrane. So here we are moving from a region of high concentration. We are moving from a region of high concentration and going to a region of low concentration. But as we are flowing in that direction, there is no expenditure of energy. No energy is required. That one is what we refer to as simple diffusion. And this one is mainly for small molecules. It is mainly for smaller molecule, or what we refer to as lipophilic molecules. They're the ones that use simple diffusion. Another type of passive transport is facilitated diffusion. Now, in facilitated diffusion, we are still moving from a region of high concentration going to a region of low concentration. And that movement from a region of high concentration to a region of low concentration, we don't use energy. But what is happening here in facilitated diffusion, here we are having large molecules or molecules that are charged. These ones, for them, they will not use simple diffusion because they are large and they are charged. Therefore, this one will require some transport to move across the cell membrane, and therefore they will be transported via the membrane proteins. We talked about the types of cell membranes or types of membranes, and we looked at the different components of the membranes that make up the cell membrane. And now we are talking about facilitated diffusion. We want materials to move across the membrane. Now, in facilitated diffusion, this one helps large or charged molecules to move across the membrane, to be transported across the membrane without using energy. But because they are very large and some of them they are charged, now they are requiring some assistance to move across the membrane. Therefore, they move via the membrane proteins. They move via the membrane proteins. That one is what we refer to as facilitated diffusion. Another type of passive transport is osmosis. And osmosis, it is water moving. So water movement is determined by the relative solute concentration. It's determined by the relative solute concentration. So that means that osmosis is a passive process. And if it is a passive process, that means that water is moving from a region of high concentration and it is going to go to a region of low concentration. And if we are talking about concentration, which type of concentration are we talking about? The concentration that we are talking about is the concentration of the water molecules. Water is moving from a region of high concentration and is going to a region of low concentration. So we can say that osmosis is the movement of water molecules from a region of high concentration, their high concentration, to a region of their low concentration. There, the concentration we are talking about is the concentration of water. But still we can state it in the other way around when we are talking about the concentration of a solute. There we can state that osmosis is the movement of water molecules from a region of low solute concentration to a region of high solute concentration. Now there, the concentration that we are referring to is the concentration of a solute. So where we have high water concentration, there the solute concentration is low. And where we have low water concentration, at that point the solute concentration is high. So I just wanted to clarify that one so that we know. Osmosis is a passive process, and passive process is always moving along the concentration gradient. And when we say along the concentration gradient, it means that molecules or substances are moving from a region of high concentration to a region of low concentration. But since this is osmosis, it is the water which is moving. And if water is moving, water is going to move from a region of its high concentration to a region of its low concentration. And where water is moving from, that region has low solute concentration. And where water is going, that region has a high solute concentration. I hope you have got that one. Now, we want now to talk about simple diffusion. I want us to understand simple diffusion. In simple diffusion, this is the net movement of particles from a region of high concentration to a region of low concentration until equilibrium is reached. So we are saying, when we say simple diffusion, it is a net movement. And what is movement are the particles. The particles are the ones that are moving, and they are moving from a region of their high concentration to a region of their low concentration. Now that one, when the particles are moving from a region of their high concentration and going to a region of their low concentration, that is what we say that they are moving along the concentration gradient. The gradient is created by particles moving from high to low region of concentration. Now, this one means that particles are moving from a region of high concentration and they are going to a region of low concentration. That is what we are referring to as concentration gradient. This movement will continue until when an equilibrium is reached. What do we mean by an equilibrium being reached? An equilibrium is reached when on both sides the one which had a high concentration and the one which had a low concentration, the number of particles are the same on all sides. For example, when we look at our diagram here, you can see that a region of high concentration, we have 7 particles. And a region of low concentration, we have 3 particles. So when simple diffusion is taking place, particles are going to move across the semi-permeable membrane, or what we are referring to our membrane here. The particles will move across the membrane, and the particles will be moving in all directions, from low concentration to high concentration, from high concentration to low concentration, until when an equilibrium is reached. When an equilibrium is reached, it means that the particles on the left side are equals to the particles on the right side. Therefore, there we shall say that an equilibrium is reached. So as diffusion is taking place, now it will reach an equilibrium. When it reaches an equilibrium, that means that particles on the left, when you count them, they are 1, 2, 3, 4, 5, will be equal to the particles on the right. That is what we are referring to as simple diffusion. Another one is facilitated diffusion. When we were talking about facilitated diffusion, we talked about the protein channels. Substances moving through the protein channels and others moving through the protein carrier channels. Now, here, what we are referring to is that certain substances cannot move freely across as a plasma membrane. Some of the substances cannot freely cross the plasma membrane. And which substances are those that we are talking about? We are talking about the ions, and we are also talking about the polar macromolecules. Those ones now, they need some facilitation because they are charged, and others are polar macromolecules, meaning that they are very big, therefore they cannot easily move across a semi-permeable membrane or a membrane via the simple diffusion. Now, in facilitated diffusion, that is now when we come in with the protein channels. We have the protein channels and then also the carrier proteins. These ones are going to facilitate the movement of these substances, which are ions and polar macromolecules. Now, the membrane proteins are the ones that are going now to do the facilitation. And this facilitation, they are going to help these molecules to move from a region of their high concentration to go to a region of their low concentration. They will move via the protein carriers and the protein channel. That is what we are referring to as facilitated diffusion. Now let us first of all look at the protein channels. Still it is a facilitated diffusion. Now for the protein channels, we have our protein is this one. This is our channel which has got protein, which we are referring to as the protein channel. Now these protein channels, they have what we call hydrophilic pores. They have hydrophilic pores to allow the passage of ions. Now when we talk of hydrophilic, we mean that this one is water-loving. And being water-loving, being hydrophilic, it will allow the passage of ions. So the ions now will move from a region of their high concentration and they will go to a region of their low concentration after passing through a protein channel. The protein channel is now the one which is facilitating the charged ions to move through the membrane and move them along their concentration gradient. Now, when we look now at the carrier proteins, now our carrier proteins here, they are also going to help the particles to move from a region of their high concentration and go to a region of their low concentration. But they are going to pass through a protein. They will pass through a protein. And now what is going to happen? This protein, there is going to be a conformation change. We are calling it a conformation change. And that conformation change is the one that is going to enable the translocation of those particles from a region of their high concentration to a region of their low concentration. Now, this conformation is taking place in the protein. The protein itself is going to change shape. That is what we are referring to as conformation change. When it changes shape, it will pick particles from the region of their high concentration and it will carry them to the region of their low concentration. Now, that one we see that the particles are being facilitated to move across the membrane from a region of their high concentration to a region of their low concentration. But this one is because these particles are charged and these particles are big. Therefore, they cannot easily move across the membrane by simple diffusion. They are facilitated by the protein channels and the carrier proteins to move across the membrane. Examples of channel proteins. We want to get examples of channel proteins. Now, we have one of them is the potassium channel. The potassium channel. Now, in the potassium channel, we have what we refer to as integral proteins with the hydrophilic inner pores via which potassium ions are going to be transported. Now, here we have what we are referring to as integral proteins. Integral proteins, those are the proteins that move across, inside, and then they move through the membrane. They move through from one side to another side, from the intra to the extra. They are moving from outside the membrane, passing through the membrane, entering inside. So, the sodium channel, it is an integral proteins with hydrophilic inner pores. Hydrophilic inner pores, those are pores which will allow ions to pass through, the potassium ions to pass through that channel of the protein. So when the channels are open, they will allow the particles or potassium ions to go through. And when the channels are closed, they will not allow the potassium ions to go through. Now, these channels that we are referring to as the potassium channels, they adopt an open stroke a closed conformation. And this one is going to be based on the membrane polarity, what we are referring to as voltage gated, or by binding of a neurotransmitter substance, or what we call the ligand gated. Now, the channels are adopting an open and a closed conformation basing on the membrane polarity. Now, here we can see that this channel here is closed. The channel is closed. And the channel on the other side, the channel is open. Now, this is because of the polarity on the membrane. So here we can see that the channel is closed, and here the channel is open. And when the channel opens, the particles now will move from a region of their high concentration and going to a region of their low concentration, because the channels have been opened. So the opening of the channel is going to be based on the membrane polarity. If the membrane becomes polarized, the channels are going to open. And the channels, when they open, they will allow the potassium ions to move from a region of their high concentration to a region of their low concentration. Sometimes this one can happen in neurons. It can happen in neurons. When a transmitter substance is released, it is going to polarize the channel up. And when the channels are polarized, they will open. And when they open, now they will allow the particles, or they will allow the potassium ions to move across the channel, protein channels and the potassium channels, and move across the membrane to the other side where the particles are in low concentration. So, structure and function of potassium channel is to facilitate diffusion in oxygens. Oxygens, these are found in a neuron. So their structure, the structure of the potassium channels, they are going to facilitate the movement of particles across the oxygen or the neuron. Now let's go to osmosis. Let's look at osmosis. Now in osmosis, we have already discussed and said that it is the water molecules that are moving. So here we can say that it is the net movement of free water molecules across a semi-permeable membrane from a region of low solute concentration to a region of high solute concentration. So here what we are seeing that water is moving from a region of high water concentration to a region of low water concentration. Now I want us to understand this one once again. Here we are saying it is the net movement of free water molecules across a semi-permeable membrane. Water is moving across a semi-permeable membrane, and it is moving from a region of low solute concentration, and it is going to a region of high solute concentration. It is going to a region of high solute concentration. Let us first of all look at our diagram below. Here we have a region of low solute concentration. This region here is of low solute concentration. And we say that where we have low solute concentration, in that region we have a high concentration of water molecules. When we say a high concentration of water molecule, we are talking about the water molecules that are free to move. Water molecules that are free to do what? To move. So if you look at this side, you will see that we have a lot of water molecules that are free to move. Now, the water molecules will move from that region to a region of high solute concentration. Now, when we say a region of high solute concentration, that one means that it has very many solute particles. If you look at this region, it has more solute concentration. There are very many solute particles this side compared to the other side. Now, if you look at this side where we are saying there is high solute concentration, you will see that there are very many solute particles. Now, when these solute particles are in this region, they will attract the water molecules. And all these solute particles that we have circled, you are seeing them that they are holding the water molecules. And when they are holding those water molecules, now we are going to remain with the few free water molecules. Now, the only water molecules that can move now across the semi-permeable membrane are these ones that are free, that are not attached to the solutes. Those are the only free water molecules that are able to move across the membrane. Now, when you look at this other side, here we have very many water molecules that are free, that are not bound to the solute molecule or solute particles. So all these water molecules on the side which has low solute concentration are free. They are free to move across the semi-permeable membrane and go to the other side where we have low solute concentration, where we have low free water molecules concentration. So that means that water will be moving from a region of low solute concentration and going to a region of high solute concentration. So here what we have been talking about here you can see that the total number of water molecules we have, they are 24 water molecules. But the free water molecules that we have, they are also 24 free water molecules. But when we go to the other side, this side you will see that the total number of water molecules that we have, they are still 24 water molecules. But the free water molecules that we have that are free to move freely across a semi-permeable membrane, you will see that they are only 4 water molecules. That makes it that now on the left side we have more water molecules than on the right side. Therefore, those water molecules on the left side are free to move across the semi-permeable membrane and go to the right side where we have free water molecule. And that one is the one that we are referring to as osmosis. Osmolality. Now, in osmolality, this is the measure of solute concentration. We are saying here that osmolality is the measure of the solute concentration. Osmolality is the measure of the solute concentration. Now, solutions can be classified as hypertonic. A hypertonic, this one has a high relative solute concentration. Hypertonic solution, this is a solution that has got a high relative solute concentration. And a hypertonic solution always gains water via osmosis. That is a hypertonic solution. So if we are talking about osmosis, before osmosis, you might be having a solution distributed like this. And in the middle, you have a semi-permeable membrane. But on the left, we have a solution or a solution that has got very many particles. This solution, this side, has got very many particles. And the other side, we have another solution that has got fewer particles. So this means that the water molecules are going to move from a region of low solute concentration where we have less particles, and they will go to a direction where there are high particles. So that means that the side where we have low solute concentrations, the side where we have low solute concentration, that is the side where we have a lot of water concentration. Therefore, that side is hypotonic. That side is hypotonic. Because it has low solute concentration, therefore it means it has a high low solute concentration. That means that it has a high water concentration. Therefore, it becomes hypotonic. Now we have a region which has got a high concentration of solutes. This region which has a high concentration of solute means that it has low water concentration. And therefore, that region, we shall call it a hypotonic solution. So, water now will move from a region of low solute concentration and it will go to a region of high solute concentration. So here it means that when osmosis starts, the water will move from a region of low solute concentration and it will go to a region of high solute concentration. Now, that is before osmosis. So here when osmosis starts taking place, we are saying that the water molecule will move from a region of low solute concentration going to a region of high solute concentration. So the hypotonic region is going to lose water going to a region which is hypertonic. So water is going to be lost from a region which has high water concentration and it will go to a region which has low water concentration. A region with high water concentration is a region that has a low solute concentration. And a region which has low water concentration is a region that has a high concentration of solute. So that's why water is going to be lost from a region which is hypotonic and the water will go to a region which is hypertonic. That's the reason why we are seeing water levels are going down at the hypotonic region and going up at the hypertonic region. Hypotonic. This one has low relative solute concentration. It has low relative solute concentration. This region here, the one we are calling hypotonic, has low relative solute concentrations compared to the other side which is hypertonic. And the hypotonic solution loses water via osmosis. The hypotonic solution is going to lose water via osmosis because water will be moving from a region of low solute concentration and going to a region of high solute concentration. Therefore, water is going to be lost from a region of hypotonic and it will be gained in the region of hypertonic by osmosis. Now, in isotonic, this one has the same relative solute concentration. Isotonic, this is where the particles are distributed equally in all directions, and therefore there is no overall net movement of water. No net overall movement of water, meaning that the particles on the left are equal to the particle on the right. The concentration of solute on the left or the concentration of water molecules on the left will be equal to the concentration on the right. When that one happens, then we say we are at equilibrium. So, osmolality. Estimation of osmolality in tissues is by bathing samples in hypotonic and hypertonic solution. If we are to estimate the osmolality, we have to get tissues, and those tissues we bathe them into what we are calling the hypotonic and hypertonic solution. Then we can be estimating the osmolality. Examples of osmotic potential. Now, animal tissues must be bathed in isotonic solution to maintain cell viability. If the animal tissues are to survive and to stay viable, they need to be bathed in isotonic solution. In isotonic solution, it's whereby we have both sides across the semi-permeable membrane have the same concentration. Hypertonic solution. These ones are going to cause the cells to shrivel or shriveling. Hypertonic solutions will cause the solution to shrivel or substances to shrivel. Here we are looking at the red blood cells. If you get the red blood cells and you put them into a hypertonic solution, you get the red blood cells, put them into a hypertonic solution. They are going to lose their water, and that water will go from the cells which are hypotonic and go into the solution which is hypertonic. So that one is going to cause them to shrivel. They will reduce in size because they are losing water. So that's what happens when you put substances in a hypertonic solution. It is going to cause them to shrivel. Now, when you put them in a hypotonic solution, what is going to happen? They are going to lysis, or they will break up, they will burst. So for example here, if we get our red blood cells and we put them into a hypotonic solution, what is going to happen? Water is going to move from the hypotonic solution and enter into the red blood cells. And what is going to happen? This one is going to cause the red blood cell to swell. And when they swell, now they will end up bursting. and that is what we are referring to as lysis. So, pressing red blood cells into a hypotonic solution, it is going to cause lysis. They are going to take in water, and when they take in water, they are going to swell, and when they swell, they are going to end up bursting, which we are referring to as lysis. So, if it is the plant cells, for the plant cells, for them, they maintain an overall fixed shape. They are going to maintain an overall fixed shape. Why? This is due to the presence of the rigid cell wall. Plant cells, they have got a fixed shape. They have a fixed shape. And what causes this fixed shape is the presence of the rigid cell wall. So when you get a plant cell and put it in an isotonic solution, an isotonic solution is the one which has the same concentration as that one of the cell. They have the same concentration. So the net flow of water is going to be the same. There will not be any movement of water across because they have the same concentration. But when you get the cell and you drop it into a hypertonic solution, what is going to happen is that the water is going to move from the plant cell and it will enter into the hypertonic solution. And when water moves from the plant cell entering into the hypertonic solution, the membrane inside the cell is going to move away from the cell wall, and then it is going to cause the cell to start becoming smaller or changing, moving away from the cell membrane and going inside. And when that one happens, there is going to be a change in the cell wall, but it is not going to be so much because the cell wall has got a rigid cell which is made up of cellulose. Now, when we get now our cell and we put it into a hypotonic solution, if you get that cell and put it into a hypotonic solution, what is going to happen is that the cell is going to take in water. The cell will take in water. Water will move from a hypotonic solution and that water will enter into the cell. So what is going to happen? The cell is going to start swelling. But when it swells, it will not burst. Why? Because as water is entering and applying pressure on the cell wall is also again going to apply pressure against the water. And this is because the cell wall is a rigid cell wall. It is a rigid cell wall. So it will be like as if the cell wall is fixed. So in plant cell, we don't expect the cell to burst because of the rigid cell wall. Let's now talk about active transport. In active transport, there is use of energy, and this energy is going to help to pump the molecules to move against the concentration gradient. So that means that the particles are moving from a region of their low concentration to a region of their high concentration. And when that one happens, it will require some energy. That means that we are moving against the concentration gradient. We are moving from a region of low concentration moving to a region of high concentration. And that one is what we refer to as active transport. When you are moving from a region of low concentration going to a region of high concentration, there you need to use some energy. You will need to use some energy. So that is what we are referring to as active transport. Now, we have what we are calling direct active transport, or what we refer to as primary. In this one, the ATP hydrolysis is used to mediate the transport. So we are going to use ATP. So what is happening here, if we look at our diagram here we have energy which is going to come from ATP. ATP is going to be hydrolyzed, and when it is hydrolyzed, it is going to provide energy which is going to help the particles to move from a region of their low concentration to go to a region of their high concentration. That one is only possible if we have energy. And that one is what we are calling primary active direct active transport. Indirect active transport, the one which we are referring to as the secondary. Now, in this one, we are going to transport coupled. The transport is going to be coupled. One molecule is moving, and then another one will also use that opportunity to move when another molecule is moving. So transport coupled to another molecule moving along the electrochemical gradient. So you may look here, you can see that now we have this particle here. This particle is moving from a region of its high concentration to a region of its low concentration. But as it is moving like that, its movement is going now to cause these particles which are here to move from a region of their low concentration going to a region of their high concentration. Because it is using that chance whereby the other particle which is moving, it is going to use that same chance so that it also moves from a region of its low concentration to a region of its high concentration. Now, that type of movement is what we call co-transport. Co-transport. One molecule is moving along the electrochemical gradient, and then the other one uses that opportunity and it also sneaks out through the channel and it goes to another region. There is another one that we are calling symport. In symport active transportation, here both molecules are going to move in the same direction. Both molecules will be moving in the same direction. That one is what we are calling symport, and it is an indirect active transport. One molecule using a benefit of the movement of another one, and then it also sneaks. If they are all moving in the same direction, then that one we call it symport. And we have another one called antiport. In antiport, the molecules are moving in opposite direction, like the one that we have here. This one, we can see this particle is moving from the other side, coming this side, and then this one is moving from this side, going the other side. That type of movement is what we refer to as antipode. Molecules move in opposite directions. Examples of protein pumps. Want to look at examples of protein pumps. We have what we call the sodium-potassium pump. Sodium-potassium pump. Now, here, 3 sodium ions attach on the intracellular binding sites on the protein pump. You have here your sodium ions, and these sodium ions, they attach themselves on the protein pump. You can see them attaching themselves on the protein pump in the intra-binding sites. These ones inside are the ones that we are calling the intracellular binding sites. So that's where the sodium ions are going to attach themselves on the protein pump. Then ATP hydrolysis changes the conformation. It is going to change the conformation, exposing the extracellular binding site. It is going to expose the extracellular binding site. So when we have ATP here, this ATP is going to be broken down into ADP. And when it is broken down into ADP, we have now outside here an extra binding site. This one here is an extracellular binding site. So it will change its conformation. Here, our 3 sodium ions are already attached to the intracellular binding site. ATP is now hydrolyzed, and when it is hydrolyzed, it will change the conformation of the exposed binding site, extracellular binding site. Now, this one, what is going to happen? Now you can see that when the 2 potassium ions now are going to attach themselves on the sides, and the ions are going to be exchanged antipodal. So here, the sodium ions have attached on this side. We have stimulated this one, and after stimulation of that one, the sodium ions will be pushed out of the cell, and now this conformation is going to be changed. It will open, then the potassium ions will come and attach themselves there. 2 of them will be attached there, and then now this one will be destroyed, and then now the conformation will change, and the protein now will push the potassium ions inside the cell across the membrane. Now that type of movement, we are seeing the sodium ions are moving out, and we are seeing the potassium ions moving inside. That type of movement is what we are calling antipode. Antipode. So the sodium pump moves ions in the neuron. Sodium-potassium pump are used to move ions in the neuron. So that is when the action potential is taking place. 3 sodium ions are pushed out, and then 2 potassium ions are pushed in. That is during the action potential in the neuron. Now let's look at the vesicular transport. We want to talk about vesicular transport. Now in vesicular transport, molecules that are destined for secretion are transported within the cell's membranous network by vesicles. So here we have molecules, and those molecules are supposed to be transported, and when they are transported, it can be within the cell membranous, within the cell's membranous network, and these ones are the ones that we are referring to as the vesicles. So inside vesicular transport is going to take inside the cell, and this transport is going to be done by membranous network of vesicles. We have the ribosome, which synthesizes the secretory proteins, and when it synthesizes them, it pushes them in the rumen of the endoplasmic reticulum. So here what we are seeing here, this is the endoplasmic reticulum, and it is called a rough endoplasmic reticulum because it has got these small tiny structures. Those small tiny structures that you see attached to it are referred to as ribosomes. Now those ones, it's where protein is synthesized. Now when they synthesize the protein, they push it through the rumen of the endoplasmic reticulum and it goes inside the rumen of the rough endoplasmic reticulum. Now, when the proteins are in the endoplasmic reticulum, there is going to be budding of the endoplasmic reticulum membrane. And this one will create a vesicle which is going to transport the proteins to the Golgi bodies. So inside here, in our rough endoplasmic reticulum, we can see that at the tip of the endoplasmic reticulum here, you can see that there is a budding. This budding here is the one now which is going to form a vesicle. And this vesicle here is coming from the budding of the endoplasmic reticulum, then it pitches off. Now inside it, inside that vesicle, that is where we have our protein. Now that protein is going to be transported and taken by the vesicle. It will come to the Golgi body. Now when it comes to the Golgi body, it is going to bind itself on the Golgi body. Now the protein is now going again to be transported by from the Golgi body, still by the vesicles, and then it will be taken to the plasma membrane. So this vesicle, which is now attaching itself on the Golgi body here, it will be now transported through the Golgi body, and then it will bulge out again here, and then it will be transported. When it is transported, it will come now and fuses with the membrane, the plasma membrane. So we are seeing that our proteins that have been produced by the ribosomes on the rough endoplasmic reticulum are now being transported through vesicles and taken to the Golgi body. Then from the Golgi body, they will be taken to the membrane where they are going to fuse, and then they will be transported outside of the cell. So, intravesicles can move materials between the cell organelles. The intravesicles are the ones that we are looking at here. These intravesicles, they are the ones that are helping the materials to be moved between the cell organelles. So here, moving from the rough endoplasmic reticulum going to the Golgi body, it is being done by the vesicle. And then also moving from the Golgi body coming to the cell membrane, it is also done by the vesicle. So the intravesiculars or intravesicular vesicles are the ones that are going to transport the materials and move those materials between cell organelles, like the way we are seeing being transported from the rough endoplasmic reticulum, taking them to the Golgi complex, and then lastly taking them to the membrane. Now we want to look at bulk transport, or what we are calling cytosis. Cytosis. In cytosis, the bilayers are held together by hydrophobic interaction between the fatty acid tails. So here what we are looking at the bilayers. We are saying the bilayers, they are held together by the hydrophobic interactions between the fatty acid tails. So here we have our membranes. So you have this bilayer here, and then you have the second bilayer here. The fatty acids are those regions in the middle. Those are the fatty acid tails. And those fatty acid tails are the ones that are interacting with each other. And they do so by hydrophobic interactions. Now, those interactions can be easily broken and reformed. So we are having those interactions there. Those interactions between the fatty acid layers can be easily broken. They can be broken, and after being broken again, they can be reformed. But for those interactions to be broken, it means that we are going to require energy. So what we are going to do, we are going to hydrolyze ATP, and that ATP is going to be used to break those interactions between the fatty acid tails and then also reforming them. Now, the separation, that separation or the fusion of the membranes allows the material to be transported. When the membranes separate or when they fuse, they are going to allow the movement of material across the membrane. So we can see here these membranes or the tails are breaking up and joining. They break up and join, break up and join. And as they are doing that, they are trying to create a way where the materials have to pass. So with the time, as they separate and they also fuse, they are creating a way where the materials will pass. And with time, we are going to see a route which is created between the membranes. And that route which is created, it's where the materials are going to pass. And that is what we call bulk transport, or what we refer to as cytosis. So, because of that movement of the materials, what is going to happen is that the fluidity of the membrane being broken down is going to allow the materials to be taken into the cell by a process called endocytosis, or the materials to be released out of the cell by the process called exocytosis. Here, when we are looking at our membrane, we started the membranes when they were existing. Then there was the breaking of the tails of the fatty acids, and then the reforming of the tails reforming and breaking. And as they are breaking, now they are creating a way where there is no way. And very soon, we are now going to have a way created. And this way which is created is going to allow the materials to enter inside the cell. And then that is what we are calling as endocytosis. And then now it will allow other materials to get out of the cell. And that one is what we are calling exocytosis. This one has been brought about by the membrane's fluidity being broken down and breaking down the membrane and creating a way for the materials to be taken into the cell, which we are calling endocytosis, and also to be released out of the cell, which we are calling exocytosis. That one is what we refer to as bulk transport, or what we call cytosis, endocytosis and exocytosis. We look at now exocytosis. Now, in exocytosis, the materials are going to be released from the cell via the vesicles. In exocytosis, materials will be released from the cell going outside of the cell, but it is going to move via vesicles. In endocytosis, the materials internalized within vesicles. The materials are internalized within vesicles. So with endocytosis, now the materials will be moving from outside and they will be entering into the cell. But they are going to do that one when they are passing through what we are calling the vesicles. Let's look at endocytosis here. Endocytosis is a process by which substances enter into the cell without passing across the plasma membrane. That is endocytosis. Substances are entering into the cell without passing through the plasma membrane. So the substances become internalized within the vesicles. So when you look here at one, we have here materials that are going to enter into the cell. They will come very close to the cell membrane. I think all of us here, we can see this structure here. This structure that you are seeing moving like that, that is the cell membrane. So when the material comes in, the cell membrane bulges inside. And when you see here, as the materials are entering inside, the cell membrane continue to bulge inside, moving inside. And very soon, if you come to number 3 here, you will see that now the cell membrane will form an inside structure. Now this inside structure is what we are calling the vesicle. But still the cell membrane is still outside here. It reforms again and it allows the vesicle to go inside. Now this vesicle is the one now that we have here. It is the one which is carrying the materials that have been transported inside the cell. That one is what we are saying, that endocytosis is a process by which substances enter in the cell without passing across the plasma membrane. So our substances were outside here and they went on entering inside without passing through the plasma membrane. They just entered, and then the vesicle was formed by the breaking, separation and the fusion of the plasma membrane. And then it formed a vesicle, which vesicle went inside, and then the membrane remained outside, and the materials were carried inside the cell or were internalized within a vesicle. Phagocytosis, still the same. In phagocytosis, this is an ingestion of solid substances, an ingestion of solid substances. Now here, the food particles, foreign pathogens are all on. They also move inside the cell using the same process, which is endocytosis. They will just enter inside, and then the membrane will bulge inside, and then it will rotate around the materials. When it rotates around the food particles, then the outside, the membrane joins together, and then the vesicle goes inside. And now those foreign materials or food particles have been taken inside the cell. And that process of taking in solid substances is what we refer to as a phagocytosis. Then we have pinocytosis. In pinocytosis, here we are ingesting, and what we are ingesting, they are liquid or solutions, not solids. They are liquid or solution. The mass intake of dissolved solutes. These ones, the substances are going inside the cell, but this time they are in liquid state. So the liquid will come, press against the cell membrane, and the cell membrane will bulge inside, and as it is bulging inside, the vesicle will be formed around the liquid, and then the membrane will form outside, and then the liquid will be trapped or transported and taken into the cytoplasm of the cell. That one is what we are calling ingestion of liquid or solution. And the substances that we are taking in are always those ones which are dissolved solute. And that one is what we call pinocytosis. So pinocytosis and phagocytosis are all endocytosis. We are taking in materials inside the cell. But the difference between pino, the substances that we are taking in the cell, those substances are either liquid substances or they are in solution state. Whereas for phagocytosis, the substances that we are ingesting, they are solid substances. Exocytosis. Here in exocytosis, This involves material exiting a cell. The materials are exiting the cell, but without passing through the plasma membrane. The materials are packaged and they are stored within the Golgi complex via excretion. So inside, we are having our materials that we want to transport. They are inside the vesicle. These materials that you see inside the vesicles here, they are the materials that we want to transport outside of the cell. And if we are transporting them outside the cell, that process is what we refer to as exocytosis. Now, those materials, we have said that the materials are packaged. And because they are packaged and stored in the vesicles, now the vesicle will move towards the cell membrane. You're seeing your vesicle moving towards the membrane. And when it reaches the membrane, now there is going to be the interaction between the membrane of the vesicle and the membrane of the cell membrane. And when they interact, there is the breaking of the hydrophobic bonds, or the fatty acids will break up and then reform and connect with that one of the cell membrane. And when they connect together, now there is going to be the release of the substances from inside the cell going outside of the cell. That process is what we are calling exocytosis. Materials have been removed from the cell, and those materials have gone out of the cell without passing directly through the cell membrane, but being facilitated by the vesicles which are moving them. And the vesicles were interacting with the membrane, and that interaction caused the materials to be excreted out of the cell. Exocytosis, the materials can immediately be released following their synthesis. When materials are released immediately after they have been synthesized, that one is what we refer to as constitutive secretion. Constitutive secretion, materials are released immediately after they have been synthesized. And when the materials are released or delayed until when a signal is given, that one is what we refer to as regulatory secretion. In regulatory secretion, materials are going to be released, but after they have been released inside the cell, they can delay until when a signal is sent that those materials are needed, and then those materials will be released out of the cell. That process is what we are referring to as the regulatory secretion. Materials are produced inside the cell. They can delay inside the cell after being produced, but when a signal comes, then the vesicle will move towards the cell membrane, and it will fuse with the cell membrane, and the materials are going to be released out of the cell. That one is called regulatory secretion, and it is exocytosis. Hello, thank you very much for participating in membrane transport. We have talked about the membrane transport. We have seen diffusion. We have discussed facilitated diffusion. We have looked at osmosis, stroke osmolality. We have looked at active transport. We have talked about vesicular transport, and we talked about the bulk transport. Thank you. Please don't forget to subscribe.